Good morning, class. Today we are going to go over chapter 14, Host Defenses 1. We're going to learn how to summarize the 3 lines of host defenses, 1, 2, and 3. We're going to identify 2 components of the first line of defense, discuss the role of normal biota as a first line defense mechanism. The first line of defense are barriers that block invasion at the portal of entry. Does not involve recognition of foreign substances and its general inaction. The second line of defense is non-specific, internalized system of protective cells, inflammation, and phagocytosis. The third line of defense is acquired on an individual basis as each foreign substance is encountered by lymphocytes. Reaction with each microbe produces unique protective substances in cells that can come into play if the microbe is encountered again. What do we call that in our immune system? The fact that you come into contact with a microbe and then your immune system does what? So when you come in contact with that same microbe again, it does what? It recognizes it, it remembers it. And it provides long-term immunity, the third line of defense. This chart, I like this chart. It specifically tells you about each line of defense. The first and second lines of defense are innate. What does innate mean? Innate. Natural ability, inborn. It's you, it's in you, it's innate. And non-specific. That's the first and second. But the third line of defense is acquired and it is specific. All right, now in the first line of defense that we're going to be talking about in this chapter, a surface protection composed of anatomical and physiological barriers that keep microbes from penetrating sterile body compartments. There are physical barriers and chemical barriers. What do you think some of those physical barriers are? Your skin. What helps keep things out of my nose? My nasal hairs. Can you think of something else, some other physical barrier that keeps, Tears. Okay, saliva's on the inside. It might keep it from going on down if you don't swallow it. All right, the second line of defense is a cellular and chemical system that comes immediately into play if infectious agents make it past the surface defenses. So if it penetrates, if it gets beyond the skin, beyond your nasal hairs, some people have little hairs in their ear. Innate response activator B cells, gamma T cells, and natural killer T cells are both involved in the second and third lines of defense. In the second line of defense, phagocytosis, what is that?
Cell eating.
Cell eating, okay. A phagocyte, which could be a what? What kind of white blood cells, okay? A phagocyte eats up microbes. Inflammation is what? Fever is antimicrobial proteins. Oh, golly gee, thank you. All right, and then our third line of defense is acquired and specific, includes specific host defenses that must be developed uniquely for each microbe. So, through the action of specialized white blood cells, and it involves B cells and T cells. So let's look at all of these. Ma'am, your saliva has lysozyme in it. And that lysozyme can kill. First of all, lysozyme is and here it is, going down your throat right here. It is a physical barrier. It is a chemical barrier or a physical barrier. Which one? Okay, all right. Now you have sebaceous glands, tears, someone already said tears, mucus. What does all that snot in your nose do? Cilia. Where is this cilia that I'm pointing to right here? You see where I'm pointing? Oh, I thought this. Because I got the light on. Okay, well, let's see this problem we have. Okay, so now can you see this cilia? Oh, well. What do we have in our stomachs? Stomach acid. Intestinal enzymes in our colon, in our small intestine and large intestine. We have mucus there. Defecation, urination. These are all, and intact skin is the biggest one. These are all physical and chemical barriers. The skin has epithelial cells that are compacted, cemented together, and impregnated with keratin. Keratin. The skin has keratin. It's a thick, tough layer that's highly impervious and waterproof. Few pathogens can penetrate this unbroken layer. Other barriers associated with the skin are hair follicles, hair shaft periodically extruded, follicle cells are desquamated. Then we have sweat glands. Did someone say sweating? It's the flushing effect of sweat glands that helps remove microbes. Mucous membranes of digestive, urinary, and respiratory tracts and of the eye. They are moist and permeable. Permeable just means what? Something can penetrate it. They provide a barrier protection without a keratinized layer. A mucus coat impedes the entry and attachment of bacteria. Blinking and tear production rid the eye of irritants. The flow of saliva carries microbes to harsh conditions of the stomach. Here in the respiratory tract, nasal hair traps larger particles. So a great big microbe or a microbe in mucus will be trapped before it gets further down the respiratory tract. Copious flow of mucus and fluids during allergies and colds exerts a flushing action. In the respiratory tree, ciliated epithelium moves foreign particles trapped in mucus toward the pharynx for removal. In the genitourinary tract, we have protection through continuous trickle of urine through ureters and bladder emptying that flushes the urethra. Vaginal secretions provide cleansing of the lower reproductive tract in females. And back to our resident microbiota. What's the other kind that we can have? Resident means it lives on us. What is the other kind we call it? Transient. Resident provides microbial antagonism, blocks access of pathogens to epithelial surfaces. It creates unfavorable environments for pathogens and competes for nutrients. It alters the local pH. Non-specific chemical defenses such as skin and mucous membranes, have lysozyme that is found in tears and saliva. It hydrolyzes peptidoglycan in the cell walls of bacteria, lactic acid and electrolyte concentrations in sweat, and the skin's acidic pH and fatty acid content. These sebaceous secretions exert an antimicrobial effect. We're going to do a lab on antimicrobials. I have a student in my other class that can't be around penicillin, ampicillin, any kind of psyllin. Does anybody in here have a problem where they know certain medications they can't be? Amoxicillin, okay. We'll make sure that you don't use that one. Remember, when we get to it, just say, I can't do amoxicillin.
We're not ingesting it all.
No,
I
this.
just can't ingest it.
Well, this girl said she can't touch it. She can't be around it. If anybody in her group has amoxicillin. So we'll work that out when we get there. Some other non-specific chemical defenses. In your stomach, you have hydrochloric acid. In the intestines, we have digestive juices and bile. We have semen and antimicrobial chemicals. And the vagina has a protective acidic pH that's maintained by normal biota. The skin produces what to create a waterproof barrier to microbes? Keratin. A thick layer of what in the nasal and respiratory passages traps bacteria? Cilia. Nasal hairs. Blank blank creates unfavorable environments for pathogens by competing for nutrients and altering pH. What is that? All right. What competes for nutrients and altering pH? Tell me now, tell me later. Tell me on the quiz. Tears and saliva contain what that hydrolyzes the peptidoglycan of bacterial cell walls? Lysozyme. The low pH in the blank and blank prevents the growth of microbes. The low pH in the what? Okay, prevents the growth of microbes. Thank you. We're going to define the word marker in section 2 and discuss its importance in the second and third lines of defense. Immunology. You've taken anatomy and you've taken 103, some of you. And so you know what immunology is. It involves our immune system. It's the study of all features of the body's second and third lines of defense. It is central to the study of the fields as diverse as cancer and allergy. A healthy, functioning immune system is responsible for surveillance of the body, recognition of foreign material, and destruction of entities that are deemed to be foreign. Pathogen recognition. We know that a pathogen can do what? What is a pathogen? Causes disease. White blood cells move throughout the body searching for potential pathogens, trained to recognize self or body cells and non-self. This spotted cell either belongs to you itself or it doesn't belong to you. Bacterial cells or other invading pathogens. Markers are molecules on the surfaces of cells. They are composed of proteins and or sugars. They are evaluated by cells of the immune system. What are 3 responsibilities of a healthy immune system? What's the immune system responsible for doing? Okay, yes. And how does it do that? By surveillance of the body, recognition of foreign material, and killing them. Destruction of entities deemed to be foreign. Cells marked as self are not marked for destruction by the immune system. True or false? Cells marked as self, these are mine. They are not foreign pathogens. These are my cells. Are not marked for destruction by the immune system. True or false? True. Markers are composed of what? What are markers composed of? Okay, proteins and or sugars. Non-self cells, such as food or commensal organisms, are attacked by the immune system. True, false? Do our cells, does our immune system attack all foods? You eat every day, right? Okay, in this section, we're going to learn to name 4 body compartments that participate in immunity. We're going to list the components of the mononuclear phagocyte system, describe the structure and function of the lymphatic system, differentiate between whole blood plasma and serum, name 6 types of blood cells that function in non-specific immunity and specify the most important function of each, and describe the major roles of T and B lymphocytes. The body compartments that participate in the immune function are the mononuclear phagocyte system, spaces surrounding tissue cells that contain ECF, extracellular fluid, the bloodstream, and the lymphatic system. What are those body compartments? The mononuclear phagocyte system, the spaces surrounding tissues that have ECF, the bloodstream, and the lymphatic system. Communicating body compartments. At the microscopic level, clusters of tissue cells are in direct contact with reticular cells and extracellular fluids. Blood and lymphatic capillaries are also present. You see the blood here, and here are the lymphatic capillaries. What color are they shown here? Like a mustard color. Yellow. Cells and chemicals that originate in the reticular system or ECF diffuse or migrate into the blood or lymphatics. So what is the flow of events that happens? The flow of events depends on where an infectious agent or foreign substance first intrudes. A typical progression would be from the extracellular spaces and reticular tissues, lymphatic circulation, and then the bloodstream. So here in this mononuclear phagocyte system, it supports the network of connective tissue fibers. Reticular system, which originates in the basal lamina. It interconnects nearby cells. It meshes with the massive connective tissue network surrounding all organs. We have a greater concentration of phagocytic cells where you see the purple here. Lymphoid tissues, GALT and MALT. GALT is gut-associated lymphoid tissue. It's lymphoid tissues in the gut. Where is your gut? Mucosa-associated lymphoid tissues, MALT, are lymphoid tissues in the mucosa. So how can we remember GALT and MALT? GALT is in the gut. MALT is in the mucosa-associated lymphoid tissue. The major functions of the lymphatic system are to provide an auxiliary route for return of ECF fluid to the circulatory system. It acts as a drain-off for the inflammatory response, and it renders surveillance, recognition, and protection against foreign materials through a system of lymphocytes, phagocytes, and antibodies. Lymph is a plasma-like liquid that's carried by the lymphatic circulation. It's made up of water, dissolved salts, and 2 to 5% protein. It transports numerous white blood cells, fats, cellular debris, and infectious agents that have gained access to tissue spaces. Lymphatic vessels permeate all parts of the body. Thin walls are easily permeated by ECF that is escaped from the circulatory system. There are two differences between bloodstream and lymphatic system. The lymph moves in one direction only, from the extremities to the heart. It's only transported through the contraction of skeletal muscles. So, tell me, blood moves in which direction? Blood. All right? It moves from the extremities to the heart and from the heart to the extremities. It goes both ways. But lymph is only transported through the contraction of skeletal muscles. Does your blood use skeletal muscles to move? No. Here's this first picture of the circulatory system, where we have body compartments are screened by circulating white blood cells in the cardiovascular system. Then we have the lymphatic system here. You have tonsils, cervical nodes, thymus, thoracic nodes, axillary nodes, the spleen, GALT, inguinal nodes. Where are these inguinal lymph nodes? All right, right in here. Then you have pelvic nodes. You have abdominal nodes and thoracic nodes. What is the lymph node called that's right here like the base of your neck? Which one is that? Okay. The lymphatic and circulatory system. Now notice, here this blood is moving from the heart down the body. We have a lymphatic duct. that travels from the extremity toward the heart. Comparison of the generalized circulation of the lymphatic system and the blood. Although the lymphatic vessels parallel the regular circulation, they transport in only one direction, unlike the cyclic pattern of blood. Direct connection between the two circulations occurs at points near the heart, where large lymph ducts empty their fluid into the veins, that is, into the circled area. See the circled area right here? Where the blue and the green intersect right there? Direct connections between the two circulations at points near the heart where large lymph ducts empty their fluid into veins. All right, organs and tissues, our lymph organs and tissues. So what are they? Organs and tissues that perform lymphoid functions: the thymus, lymph node, spleen, and malt. TLSM. Thymus, lymph node, spleen, and malt. A loose connective tissue framework that houses aggregations of lymphocytes. The thymus is the site of T-cell maturation. It's a triangular structure in the pharyngeal region. It's largest at birth proportionally. So that means a baby has a large thymus, don't they? Compared to how large your thymus is once you get to be an adult. It exhibits high rates of growth and activity and growth until puberty. It shrinks gradually through adulthood. The thymus. What happens in the thymus? What kind of maturation? T cells. Lymph nodes are small, encapsulated, bean-shaped organs. They are stationed in clusters along lymphatic channels and large blood vessels. There are major aggregations that are loose connective tissue in the armpit. Do you have a lymph node in the armpit? The groin, the neck. It's ideal for filtering out materials that have entered the lymph. The spleen is a lymphoid organ in the upper left portion of the abdominal cavity. It serves as a filter for blood instead of lymph. Its primary function is to remove worn-out red blood cells from circulation. And it has an immunologic function where it filters pathogens from the blood for phagocytosis by macrophages. People with sickle cell disease have a lot of worn out red blood cells that need to be taken from circulation. I have a little cousin with sickle cell whose spleen was the largest this doctor had ever seen. What does that say about his red blood cells? He's got a lot of them that are worn out. They're in his spleen. What is the primary function of the spleen? To remove them. So is his spleen working well? If he has the largest spleen a doctor's ever seen, something's not working right, is it? Miscellaneous lymphoid tissue. Bundles of lymphocytes beneath the mucosa of the gastrointestinal and respiratory tracts. That's where we have our tonsils, the breast of pregnant and lactating women, galt, malt, salt, and balt. Anybody choose to take a guess at salt and balt? We know galt and malt, right? Galt, gut-associated lymphoid. Malt, mucosa. What's salt and balt? Let's learn it. Peyer's patches. We also have Pierce patches associated with lymphoid tissue. Here is the composition of whole blood. Blood cells suspended in plasma. Plasma is the clear yellowish fluid. See right here where I'm pointing? Plasma. Serum is the same as plasma, except it contains no clotting factors. Serum right here. It's used in immune testing and therapy. Plasma and serum. Blood cells are suspended in plasma. Components of plasma. It's 92% water. It has protein, albumin, globulin, and antibodies. There are other immunochemicals in plasma. Fibrinogen and other clotting factors. Fibrinogen. Hormones, nutrients, such as glucose, amino acids, and fatty acids, ions like sodium, potassium, calcium, magnesium, chloride, phosphate, and bicarbonate. It has dissolved gases, oxygen and carbon dioxide, and waste products include urea. So there are hundreds of different chemicals produced by the liver, white blood cells, endocrine glands, and nervous system that all are components of plasma. Hematopoiesis is the production of blood cells. It begins early in embryonic development in the yolk sac. It's taken over by the liver and lymphoid tissues, and it's assumed permanently by the red bone marrow. Stem cells are precursor to blood cells. It's a pool of undifferentiated cells. It's maintained in the bone marrow. Its immature or unspecialized cells develop the specialized form and function of mature cells. Why do scientists want to use stem cells in their research and development of whatever they're working on? Why not just use adult cells? Because they're not differentiated. They can make them cells of the whatever they want them to be. So here, this chart tells us the development of blood cells and platelets. Here we see red blood cells. Here we learn white blood cells, leukocytes, are categorized by staining characteristics. Neutrophils, basophils, eosinophils, monocytes, lymphocytes, and within there we have our B cells, T cells, and NK, natural killer cells. We have mast cells, dendritic cells. See the little dendrites hanging off of them there? More in depth. White blood cells, leukocytes. They are evaluated by reactions to hematologic stains that contain a mixture of dyes. They are differentiated by color and morphology. What is morphology? They appear with or without colored granules in the cytoplasm. Granulocytes have a lobed nucleus. Agranulocytes are unlobed and have a rounded nucleus. What does the letter A in front of a word do to any word? Okay, without. It negates whatever the meaning of that word is. So granulocytes are neutrophils, eosinophils, and basophils. They're prominent cytoplasmic granules that stain with eosin or methylene blue. The granules function in numerous physiological events. Neutrophils, eosinophils, and basophils. Neutrophils are 55 to 90% of the circulating leukocytes. We have production of toxic chemicals and phagocytosis performed by neutrophils. Eosinophils, granules contain peroxidase, lysozyme, and other digestive enzymes, toxic proteins, inflammatory chemicals. They attack and destroy large eukaryotic pathogens. Eosinophils, that's what they do. Basophils make up less than 0.5% of circulating white blood cells. They share morphological and functional characteristics of mast cells. So, of the neutrophils, eosinophils, and basophils, which one makes up the least amount of white blood cells? Basophils. The largest one would be the neutrophils, 55 to 90%. Agranulocytes, meaning they are not granulocytes. So they are globular, non-lobed nuclei. They lack prominent cytoplasmic granules. There's two different types, lymphocytes and monocytes. We just got through. All right. Two general types, lymphocytes and monocytes. Lymphocytes are the cornerstone of the third line of defense. They comprise 20 to 35% of the circulating white blood cells. It is estimated that 1/10 of all adult body cells are lymphocytes. They are exceeded only by the red blood cells and the fibroblasts. They have three functional types: B lymphocytes, bursal equivalent; T lymphocytes, thymus-derived; and null cells. Activation of B lymphocytes. It's an antibody-mediated immunity. It's also called humoral immunity. B lymphocytes activation is also called humoral immunity. They form plasma cells that produce antibodies. What are antibodies? Large protein molecules that interlock with antigen and participate in their destruction. Antibodies participate in the destruction. T lymphocytes, T like Tom, produces cell-mediated immunity. It's a wide spectrum of immune functions. They modulate the immune functions and kill foreign cells. That's why they're called killer T cells. The action of both cell types accounts for recognition and memory, typical of immunity. So what kinds of cells are responsible for recognition and immunity? B cells and T cells. All right, I think that we'll pick up the next time with monocytes and macrophages. Stay tuned for part 2.